Okay, so we know the characteristics of a generic cell. Plasma membrane, some sort of DNA, cytoplasm, and ribosomes. Well, now we're going to compare different cell types. First up is going to be what is referred to as the prokaryotes. Prokaryotes, better known as the bacteria. Prokaryotes lack any membrane enclosed organelles. No nucleus, no Golgi, ER, any of those other things you've learned in high school. One of the things we're going to see with most of the prokaryotes, they're going to have some sort of thing referred to as peptidoglycan. Peptidoglycan forms a cell wall. It's a solid structure, somewhat solid structure, composed of peptides, proteins, and glycans, sugars. Sugars and small polypeptides are going to form this somewhat, how do I phrase this? It's going to form this cell wall that is somewhat rigid. We believe that prokaryotes that we see nowadays are quite similar to what's been around since the beginning. Prokaryotes are believed to be the first life forms on this planet. Think about it. You're going to evolve plasma membrane, some sort of a membrane, lipid bilayer. You're going to have some sort of nucleic acid, maybe a small chunk of DNA, RNA, some enzymes and ribosomes. Boom. Well, right there you go. You've got a bacteria, a prokaryote. May not have the cell wall and all the other stuff that modern day bacteria do, but it meets our definition of a prokaryote. When you look at a prokaryote, they're not all going to have all of the things shown here by the artist. This is just a generalized, ooh, look, here's some of the things you could find. What you will find, a circular strand of DNA that's free floating around in an area called a nucleoid. Okay, it's not a nucleus. There's no membrane. It's just a region in the cell where it's all kind of just put there. There's going to be ribosomes. All living cells have ribosomes. There's going to be a cell membrane, a lipid bilayer, and the possibility of other things. Some sort of a cell wall made of that peptidoglycan. Outside of that, there could be what's called a capsule. Capsule is going to be a bunch of sugar moieties, long strands of sugar that hold in water, make it a slime. Or it could be something rigid, kind of like the chitin thing, remember? The insects. Okay, it's not chitin, but it does subtly the same thing. It gives it a rigid protective coating. There can also be what's called pili and flagellum. Pili are these short little protrusions that are outside the cell that, can allow the cell to stick to things. Flagella, this long whipcord-like thing, long whip, is used for propulsion, to push or pull this whole cell through liquid. A lot of times, since I'm the resident microbiologist, I get students ask me, why are prokaryotes so small? Well, in a nutshell, they have to be. Okay? They do not have mechanisms for transporting and moving around nutrients. So they're always at the mercy of their environment. It has to be some form of sugars, amino acids, and whatnot. So they're always on the edge of starvation, the cell's starving. Even if you put them in a highly enriched environment, they're not going to get much bigger because they can't pump in enough. They can't bring in enough sugars and amino acids to get bigger. So they don't have the genes, the instructions to be bigger. textbook talks about this whole surface area to volume. The bigger you become, the more surface area you have, or the more volume you may have, but you may not have more surface area. Surface area allows you to absorb more nutrients, allows you to bring stuff in. It's also, on the flip side, why we're not one big cell. It's unsustainable. By having us be, as you see here, stacks and stacks of little cells, you can have blood vessels running between them, bringing in nutrients into the deep tissue. Because if this was one big cube, not, what was that, 9 times 3, 27 cubes, you couldn't get nutrients from this side to transport all the way over to that side within the cell. It wouldn't work. Which is why when you look at the eukaryotic cells, our cells are much bigger, anywhere from 10 to 100 times bigger than a prokaryotic or bacterial cell. If you look at it, you have a plasma membrane surrounding, and you have compartments inside. Think of it as rooms. Each compartment, each room, each organelle has a different role. Inside of it's going to be different enzymes, different environments. Some parts of the rooms are going to be acidic, others are going to be basic. Some are going to have lots of amino acids, some aren't going to have any amino acids. Sugars, high, low, the whole works. So it allows our cells to move, to specialize. Not only specialize and become part of a specialized tissue, but different organelles within the cells will specialize for specific reactions. Now, the rest of this chapter, we're going to be going through the different parts of eukaryotic cells, animal and plant cells. First, going to have to talk about that plasma membrane. Last chapter we talked about phospholipids. We showed that the phospholipids has a glycerol phosphate head, and these twin tails are fatty acids. It's just a state of nature. You dump about a bunch of fatty acids into water, they're going to form this lipid bilayer. Water out here, water underneath, but no water in the middle. Why? Fatty acid tails, these chains, exclude the water. They want no water around them, whereas the heads, the round parts, want to be touching water. They want to be involved with water. But the thing is, as we're going to see in a later chapter, these phospholipid bilayers really don't do much. They're there. They're a barrier. Some things can get through, most things can't. So you have to have all of these blue things, these proteins present that allow the membranes, allow the cells to move stuff in and out, allow it to, transport, to move things, for the cell to be able to not only recognize, because some of these proteins are going to be receptors, recognize when signals are floating by in the bloodstream, going to be able to recognize and react. The cells in a tissue, the tissues of an organ have to be able to react to changes, things floating by in the blood. The cytoplasm. Remember, this is an aqueous-based solution. It's mostly water, but it's kind of thick, almost a thickish water, a thin gel. The reason being is you want stuff to float. Think about it. Last time you jumped into a pool or a lake, some people floated to the top, some people sank. You tossed in different things, some things floated, some things sank. Nothing just held in place. It either went all the way up or went all the way down. Gravity, buoyancy. So every time you move, if it was just water, organelles would float up or sink, and it would keep moving every time you moved around. Well, Since it's not completely water, it has a consistency that's semi-solid, that's almost gel-like. Things float in space. They don't float up, they don't sink to the bottom, they just hang there. No matter which way's up, no matter which way your cells are oriented. That's why we're not bags of water. Someplace near the center of the cell, you're going to have what is referred to as the nucleus. The nucleus is the organelle that is responsible for protection of the DNA, protection of your chromosomes. Generally, there's only going to be one per cell. There are some special cell types out there in the animal kingdom and the fungi that have multiple organelles. For us, that's not going to happen. If it does happen, that means you are sick with something like a RSV, a respiratory syncytial virus. It means your cells are all fusing, and yeah, it's not going to work because the nuclei would be working against each other. So, for the most part, us animals, especially us mammals, one nucleus per cell. You get down into plants, you get down into some fungi, most of the fungi, that can be different. The nucleus itself is rather large because remember, for us humans, 23 pairs of chromosomes, 46 total. Sometimes during the cell cycle, during certain times, that cell is going to have double that. Instead of 46, it's 92 chromosomes. Has a bunch of enzymes and other things going on in there. So it's rather large. Now, the nucleus, its lipid bilayer is referred to as the nuclear envelope. The nuclear envelope is dual lipid layers. So you literally have 4 layers of phospholipids, 2 membranes, one wrapped around the other. Dual protection because, well, the DNA is that important. You want to limit what can get through. If one membrane can't keep it out, 2 definitely will. But you have to have access. You have to have a way to get in and a way to get out. Ingress, egress. So you're going to have nuclear pores. The nuclear pores are special donut-shaped openings. They open and they close. Donut opens, things move in, things move out. Donut closes, nothing passes through. Within the nucleus, you're going to have the chromatin, the double helix wrapped around proteins. You're going to have an area known as the nucleolus, where that chromatin is really packaged down into thick chromosomes. The more it's packaged, the more protected it is. Now you see here, this picture shows what's referred to as the endoplasmic reticulum. It's another organelle that is attached to the nuclear envelope, and we'll talk more about that in a minute. The nuclear envelope, as I said, it's dual membranes, an inner membrane and an outer membrane. So important, evolution wrapped your DNA twice. You have these donut-shaped things, the nuclear pores that open and close due to specific proteins that are interacting with them. Messenger RNA out, other things come in. But not everything. By having the dual membranes, by having the nuclear pores that only respond to certain signals, the nucleus can limit what has access to the DNA. The nucleolus, it's an area that has lots of proteins, lots of things there that are compacting down the chromatin into chromosomes, protecting it away. Turns out there are certain genes that you only need once in your life. When you're 10 cells in size, 100 cells tell you up, down, left, right, backwards, forward, so that you can have your head up, your arms, 1 to each side, 2 legs down the bottom. Those genes, you don't want them anyone, you don't want them to be expressed ever again. So the DNA that those are on, packaged away. You're going to find those in the nucleolus. Another thing you're going to find in all cells, ribosomes. Remember previous chapter when we were talking protein structure, I said that ribosomes are made of two parts, large subunit, small subunit. You look at them right here. Large, small. Kind of in a way, if you look at it, you squint real hard, it looks like a hamburger bun. Small subunit helps to position the large subunit. The large subunit does all the work. Goes, remember, codon by codon, reading every 3 nucleotides of the messenger RNA, adding on a new amino acid, depending upon what the messenger RNA says. All living cells have these. Turns out, ours are just bigger than bacterial. Another thing you're going to find in all eukaryotic cells is the mitochondria. Mitochondria, powerhouse of the cell. You've heard that since you were a kid. The mitochondria is an organelle that houses specific enzymes that are designed, that have evolved for oxidative phosphorylation. The taking of pyruvate, remember, glucose gets cut in half, glyceraldehyde, which again gets turned to pyruvate. Pyruvate enters into the mitochondria. It powers the breaking down of that 3-carbon thing, powers reactions that allow for the making of a lot of ATP. A lot of the enzymes are going to be here on these folds of this inner membrane called the cristae. Imagine you have a bag, small bag. You have a big coat. Somehow you want to shove that coat into that small bag. You can do it. You just got to keep shoving and putting. Well, that's what we have here. The inner membrane is actually much, much, much larger than the outer membrane, but evolution has allowed for it to be shoved in there. So you get all these folds as things get compacted in there. Why do we want this? Well, real estate. Look at all those cristae, those folds. Now, granted, the artist here didn't draw that many, but there's a lot more of them in real life. Think of it as real estate. That inner membrane, that real estate, gives you lots and lots of places to put enzymes. Enzymes that are going to be there for the making of ATP. Some of the cells are going to have peroxisomes. These are little miniature bubbles inside of the cells. Bubbles that are going to have the lipid bilayer holding stuff in. Think of this more as a means of storage. That crystalline core has something in it that the cell wants to just package away. Doesn't want it reacting, doesn't want it free floating around, so it just puts it in a peroxisome. And there it's going to stick. Well, the example the textbook uses is the peroxisomes that are known for detoxifying poisons. So there's going to be enzymes in there that are going to take a poisonous substance, twist, tweak, cut, paste part of it, make it non-poisonous. That's their one and only job. Don't want those free floating around, so you put them in a peroxisome. Nice, safe place to store it.